Okay, so chapter 3 will be the transcription. Okay, in the previous chapters, we discussed those DNA-based cellular activity like replication, and that replication is relevant to the genome. So we discussed the genome, and now another DNA-based cellular activity will be the gene expression. And the first step of the gene expression is the transcription. Okay, and the next chapter we will discuss the translation, which is the next step of the gene expression. Okay, so for gene expression, let's watch this. From DNA to a protein.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones.
So we just discussed this histone, right? DNA 2 nanometer wrap around the co-histones, all right, to form this nucleosome 11 nanometer, all right, and then they fall into 30 nanometer, and then 250, 700, 7000, and then the 1400 nanometer, like this.
Within the DNA are sections called genes. These genes contain the instructions for making proteins.
And the genes are the non-repetitive DNA sequence in the genome, okay? Non-repetitive DNA sequence in the genome.
When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a...
During the DNA replication, because there's a DNA synthesis, so you need a DNA polymerase. And now here it's the RNA synthesis, so we use the RNA polymerase.
Strand of messenger RNA out of 3 bases in the nucleus. The DNA code determines the order in which the free...
So as you can see from the video, when they open that double helix, only one strand is used as a template, all right? The other one is not used, okay? So it's different from the DNA replication.
Bases are added to the messenger RNA. This process is called transcription.
Okay, so that's what we're going to discuss now, transcription, okay? Because the next step will be the translation, and that will be the next chapters, okay? So before we start this the transcription to transcribe DNA to the messenger RNA, this is just a experiment that to prove the presence of messenger between the DNA and the protein. Okay. So basically, what they did is they want to determine, they want to prove the intermediate carrier, okay, is needed, right? And because at that time, the scientists already know the DNA and protein, but they don't know the presence of messenger RNA. And then to prove this, so basically they grew the bacteria, E. coli, for several generations on the synthetic media that has a heavy isotope, the nitrogen 15, right? And also carbon 13. And carbon 13, that's a glucose carbon 13, they use it, carbon 13, and then nitrogen, right? And so the bacteria, they use this heavy isotope, the carbon and nitrogen, for the raw material to synthesize the carbohydrate, protein, and nucleic acid, right? And all these components will be the heavy labeled, all right? Including the ribosome, okay, the ribosome. And then they change the genetic instruction by infecting the bacteria cell with the T2 virus, with the virus, Okay, they infect with the virus. All right, and so basically, the virus, they destroy the bacteria DNA and then substitute their own DNA as the genetic information. And so that the bacteria will use that to direct the synthesis of viral protein. So if the ribosome carry the gene-specific information, then the new virus-specified ribosome should be met as well. Okay, if the specific ribosome is not required, the new specific ribosome is not required. If the ribosome are passive site of synthesis, then the old bacterial ribosome can be used by the virus. All right, and so and then to make the virus direct protein. All right, and then the distinction would be the concern, the source of information in protein synthesis. Okay, which occur on the ribosome. And then so basically during this process, they after this they collect the cell and then run the centrifugation, and then if there's the new ribosome, then the phage RNA should line up with the lighter one, okay? Because originally you use a heavy isotope for bacteria to grow, and then you switch, right? And then you have this regular phage to infect the cell and grow in the regular media. And then if you have the new ribosome and the new RNA, then they will be different from the old one, all right? However, when they perform the centrifugation, they found that the new RNA actually line up with the old ribosome position. So that suggests there's an intermediate that used by the old ribosome to synthesize the protein. So that's basically the very early day. That's how they interpreted the presence of messenger RNA. And then from the video that we learned that during the transcription, they only use one strand as a template to synthesize RNA. Okay, so that gives us these two strands of DNA. One is called coding strand, the other one is called template strand. So during the transcription, they use the template strand, right, to synthesize the messenger RNA, and messenger RNA will be exactly the same as the coding strand, except the T has been replaced by U. All right, so that's the template strand, coding strand. So the coding strand, template is non-coding strand, okay? And normally we also label this as a plus strand and minus strand, all right? And this is just the video about transcription.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands.
So that's the actual speed for the transcription, very fast, okay? So this is a messenger RNA. So that's a video just to show the transcription process. So it's a transcription unit, basically is the region of the promoter to the terminator, all right? And the promoter, of course, that's the place where the RNA polymerase bind. That's called promoter. And transcription start site will be actually the first nucleotide they transcribe, all right? And the first nucleotide they transcribe, we always designate as a +1, all right? And the nucleotide before the starting point is a -1, okay? And from +1, everything before +1 will be upstream. Downstream will be after +1. And terminator, there will be a signal to tell the RNA polymerase where to stop and dissociate from the template DNA. So that's the transcription unit. Okay, and eukaryotic cell, all right, basically this occurs inside the nucleus, all right, and then so we have this DNA being transcribed to the pre-messenger RNA, and then the pre-messenger RNA will be processed, and then become the mature RNA, which we'll discuss later. Yes, Marina?
I have a question about the speed of the DNA polymerase. What determines that speed? And was it always that fast from like an evolutionary standpoint?
Yeah, it's very fast. Normally, 1 minute they can synthesize like around 2 kb, 2000 base pair. Yeah.
So it was always a fast process. It was never something that solely adapted.
No, very fast process. Yes.
Okay. Thank you.
Okay. You're welcome. So then these eukaryotic messenger RNA, after they've been processed, then they'll be transported to the ribosome, and then they can translate into the protein. Okay, so that's eukaryotic process. So the RNA polymerase, they also catalyze the phosphodiester bond formation, all right? So basically again, so these, all right, so they have these to remove the pyrophosphate and connect this oxygen to the next phosphate group, all right? The same process as DNA replication, okay? And so some feature for the RNA synthesis, so our DNA template sequence determine RNA sequence. All right, so they require ATP, CTP, GTP, and UTP. And it grows in 5' to 3' direction. And one important thing is RNA polymerase does not require the RNA primer. Remember the DNA polymerase, when we perform the DNA replication, we need the primer, RNA primer. But for RNA synthesis, we do not need any RNA primer, okay? And then the numbering rule, as I say, the first nucleotide they transcribe will go for the +1, right? And then seen below that downstream, the +2, +3. And then before the -1, -2. All right, so remember this numbering, all right? Plus, minus. And this is relevant because here, the promoter, all right? So for the prokaryotic cell, the consensus sequence, all right, the strong promoter, basically there are two signals. One is -10, the other one is -35. Right, -10 and -35, we have the Tata box. All right, these are very important, okay, because these are the signal to tell the RNA polymerase where to bind so that they can initiate the transcription. So that's a prokaryotic cell. And eukaryotic cell, so when later on we will discuss, so there are 3 different types of RNA polymerase, okay, and then each type of polymerase, they have different type of promoter. But for polymerase 2, right, RNA polymerase 2, that's the one responsible for the messenger RNA synthesis. So normally they will have this -25, okay, -25 Tata box here, -25. Also, they have this proximal distal elements, and then they have enhancers, okay? So these are to help the specific transcription factor to bind, and they can stabilize the RNA polymerase 2 and to initiate the transcription. Okay, and then for this prokaryotic cell, okay, apart from this -10, -35, the core promoter, they also have this UP up elements. The up elements basically upstream this -10, -35, all right? So upstream of this core promoter. And this we call extended promoter, all right? So these from -40 to -60, that's the up elements. And this element can be recognized by a specific subunit in the RNA polymerase, which we will discuss later, okay? to enhance the transcription efficiency. So here in RNA polymerase, normally there are several different subunits, okay, where alpha subunit, beta, beta prime, omega, and sigma. These 5 different subunits, okay. And for the RNA polymerase basically include the alpha, beta, beta prime, and omega, and sigma. That will be another one that will join to the coenzyme to form the complete enzyme. We call it holoenzyme. And because sigma is responsible, sigma subunit is responsible to recognize this -10, -35. Okay, and for this up element, that will be alpha subunit. Okay, alpha subunit. So for the RNA polymerase, where coenzyme, that will be 2 alpha, beta, and beta prime. And then when we add a sigma, right, that's holoenzyme. Normally we don't discuss too much about omega. Omega is also an accessory role in the polymerase, okay? And for the coenzyme, basically alpha, alpha, beta, beta prime. And then when you add a sigma, that'll be holoenzyme, all right? Holoenzyme is very important for the transcription initiation because they need to recognize the promoter -10, -35. And then once they initiate the transcription, then the sigma will dissociate from the RNA polymerase. Then that will become coenzyme. Then the coenzyme will continue the transcription elongation until the transcription termination. And the role factor, basically, it's important for the transcription termination. Okay. So this is to show you that the role of the sigma factor in transcription, because they can induce a major change in the affinity of the RNA polymerase, okay, for the DNA. When they bind, that become the holoenzyme, all right, and then so they have they change this the RNA polymerase affinity, and they only recognize those specific promoter sequence -10, -35. Okay. And this is just to show the sigma basically initiate the transcription, then they will dissociate, all right, and then the coenzyme will continue the elongation, and this sigma will bind another free coenzyme, become the holoenzyme, and then bind to the promoter and initiate transcription. From this curve, just to show you, if you have the equal amount of a certain amount of the sigma and the coenzyme, and then after to initiate the transcription, initially the RNA chain initiation increase significantly, and once they reach the plateau, then they just don't enhance anymore. However, if you add coenzyme, then they can enhance the transcription product. Okay, that just tell you this sigma can be reused. All right. So the presence of sigma, okay, they enhance the binding of the coenzyme to the promoter DNA. Okay. And then, so of course, the transcription, we have the initiation, elongation, termination. All right. So for the initiation, basically, the RNA synthesis begin at promoter, right? When they are, first of all, when they come to the promoter with the sigma factor, right, they will find their promoter region of -10, -35, right? They form this closed promoter complex. They stop there, right? They pose, and the next step, they open up the double helix, right? Open up double helix, then you have the open promoter complex. Once you have open promoter complex, then you will incorporate the new nucleotide, few nucleotides. So you start a transcription, all right? Start a transcription around 9 to 12 nucleotide long. Then this whole RNA polymerase will move. Once they move, all right, and this stabilized, if the RNA DNA hybrid stabilized, then they move and then sigma dissociate from the coenzyme, right? And so that'll be, then they move to the next few nucleotide, and that would be the promoter clearance. So from the closed promoter complex, open promoter complex, synthesis, clearance. Okay, these are the 4 steps for the initiation. And once they enter into the clearance, then they enter the elongation. Okay. And transcription bubble, I mean, you need to open up double helix so RNA polymerase be able to synthesize the RNA. And then this region where you have this open double helix DNA, we call it a transcription bubble. All right. And for the this is just experiment to demonstrate that at this stage, all right, at this stage, how many nucleotide has been synthesized before the promoter clearance. And they use a chemical, okay? A chemical is dimethyl sulfate, DMS, dimethyl sulfate, because the dimethyl sulfate basically will transfer the methyl group to adenine, all right? So from here, so ANT, if you melt this, all right, and then you add the DMS, dimethyl sulfate, then they will transfer methyl group to this A adenine, all right, and therefore this AT will not be able to form the hydrogen bond anymore. Okay, so that's the principle. So now the experiment is that they took the double helix DNA and bind with the RNA polymerase. All right, when they bind the RNA polymerase, so the closed complex and then open complex, so they open the double helix. At this point, all right, they add the DMS, dimethyl sulfate. Then dimethyl sulfate basically transfer the methyl group to the A. Once they transfer to the A, then you remove the polymerase. They will reform the double helix. However, the place where they have methylated A will not be able to form hydrogen bond anymore. That will give you like this, right? And then you treat this DNA with the S1 nuclease, which means this S1 nuclease, they will attack this single-stranded region without the hydrogen bond. So they will cleave A here, cleave T here, cleave A here, and then you will produce different lengths of the DNA. And then when you run the electrophoresis, then you will see different lengths of DNA give you the signal in the region, and that will indicate the region where the RNA polymerase synthesize the DNA. Okay, and so that's how we see this. See this DNA here. This is from the -9 to +3 region. All right, the length of the DNA is basically give you the signal here. So that basically the length of these different lengths of the DNA give you this signal here, so that you identify that's the region where the RNA polymerase bind, and then to initiate the transcription. Okay. And this is example experiment to demonstrate the alpha subunit to recognize the up element. So they have this wild type, and then you have alpha mutation. So with the wild type, you see the up element, they can recognize up element, enhance the transcription. Without alpha subunit, you see the transcription is down. All right, so that just tell you how they determine the role of the alpha subunit in identifying the up element. Okay, and this is just the 4th step that we just mentioned. All right, so you form the closed complex and open up double helix open complex, synthesize the nucleotide, and then the clearance. Okay, transcription. And once you initiate the transcription, then the next step is elongation. So elongation basically that involve this the coenzyme. Okay, a coenzyme. So we have alpha and beta, right? And so basically the phosphodiester bond formation involve the beta and beta prime subunit. Okay, and then for the topology of elongation, so basically remember during the transcription, you're going to open up double helix. When you open up double helix, you have to rotate the DNA. When you rotate the DNA, you introduce the supercoil to the DNA. So when the RNA polymerase process the transcription, then you will introduce a positive supercoil ahead of the transcription, and also behind the RNA because after they finish this synthesis in this region, they will rewind into double helix. When they rewind, then they introduce negative supercoil. Therefore, you need the topoisomerase to remove these tension. Okay, positive supercoil ahead of RNA polymerase, and negative supercoil in after the transcription. Okay, so the topology. And during the transcription elongation, the RNA polymerase frequently pause and backtracking. So pause, that means they will allow the ribosome to keep pace with the RNA polymerase, because in the prokaryotic cell, as soon as you synthesize a new messenger RNA, then the ribosome binds and then they start the translation immediately. And backtracking basically is for the proofreading. Okay, backtracking, and also to let the raw factor, and pausing also let the raw factor to have the chance to catch up and to terminate the transcription. Okay, and then for the transcription termination, there are two types of termination. One, the intrinsic terminator. So the intrinsic terminator, that means in the DNA sequence, right? So they have this GC reach sequence, GC reach region, and also the poly T tail here. All right. So once they transcribe this region, all right, once they transcribe this region, the messenger RNA, they will have the GC reach and then poly U terminus. And then this GC reach region, they will form this secondary structure. Okay, we have stand and loop, the hairpin. All right. So once they form this secondary structure, all right, then the RNA polymerase will stop and then dissociate from the template. So this is intrinsic terminator. So it's a raw independent terminator because it does not require the raw protein. Okay, but they have a GC rich sequence and the poly U, right? And another one is a raw dependent termination. So basically, for the raw dependent termination, they have the similar GC rich sequence, but they do not have the poly U terminus. All right. For the raw dependent, they do have the GC rich sequence, because once the polymerase reach to that area, all right, they will form the secondary structure. Then the polymerase will pose. Once they pose, the raw factor will catch the polymerase, and then the polymerase then will dissociate from the template, and that's how they determine and how they terminate the transcription. And in this diagram here, just to let you know, this sigma factor should not be here, right? Because during the elongation, that should be only the core enzyme. So that means no sigma factor. Okay. So let's watch the video briefly. Oh, there's no sound. So initiation, you can see the initiation, closed complex, all right, and then open complex, all right, and then synthesis, you have transcription bubble, then you have synthesis, all right, a few nucleotide. okay, and then elongation, all right? And then as soon as you have this, the messenger RNA, then the ribosome come, and then they synthesize the protein right away. Okay. So that's the prokaryotic transcription. All right. So for the prokaryotic transcription, as we know that in the genome, let's say for example, the E. coli genome, there has more than 3,000 genes. Okay. So it is not possible for the machinery to transcribe all genes at once. Okay, so they must have a control. Okay, some gene may be active all the time, all right, because they are housekeeping genes, so they have to be transcribed all the time. Some gene must be regulated, okay, because the gene expression is very expensive process, and then for the prokaryotic cell, the bacteria, they have to be very economize all the biochemical activity in the cells. So they have to choose at the certain point, they have to use a certain protein, then they will decide to transcribe the certain genes, all right? And that will come into this regulation of the prokaryotic transcription, all right? So there are many strategy, and the 7 major strategy will be the alternative sigma factors, RNA polymerase switching, anti-termination, enhancer, chaperone, transcription attenuation, and ribo switch. All right. So alternative sigma factor, RNA polymerase switching, and anti-termination involve the change of transcription machinery. Okay. So for example, the alternative sigma factor. So for example, the regular sigma factor is sigma 70 under normal circumstances. All right, the bacteria E. coli use sigma 70 to direct the RNA polymerase for the transcription. However, when they face the environmental insult, when they involve the environmental stress, then a set of special gene will be expressed. We call heat shock gene will be expressed to protect the cell. In this situation, the heat shock response in E. coli is regulated by the specific alternative sigma subunit, which is a sigma 32. They will displace the sigma 70 and direct the RNA polymerase to the heat shock gene promoter. Okay, so that's the one example. Okay, one example, alternative sigma factor. And also they have an anti-sigma subunit. So for example, when the cell, they are stressed by insult as loss of a nutrient, high osmolarity, or high temperature, they stop growing and enter the stationary phase. So at this point, there's a protein we call RSD protein that will be produced that will bind to the sigma 70 subunit and then block the sigma 70 to bind to the coenzyme. All right. Once the coenzyme, the sigma 70 cannot bind to the coenzyme, then they stop the transcription. All right. So that's anti-sigma subunit. So these are two typical strategy E. coli use, all right, to regulate the gene expression. And then another one here, the alternative sigma factor, that'll be when the phage infect the bacteria cell, okay? Because when the phage infect the bacteria cell, they are going to direct the bacteria host to synthesize the viral protein and viral genome, right? And then so they must produce something to dominate the polymerase. Okay, so in this case there'll be a virus. It's called SPO1 spore1 virus. They can infect the Bacillus subtilis. All right, Bacillus, and so they have this set of genes called early gene. Right, the early gene, when they infect the Bacillus, right, the Bacillus will transcribe, okay, this early gene, the early transcript and early protein, right? They will transcribe this, use the the sigma 70 from the bacillus and directly RNA polymerase to synthesize early genes and produce early product. One of the products called GP28. GP28 basically is a sigma subunit. And this sigma subunit later on will direct the bacteria coenzyme to express a specific viral gene, okay? And this gene we call middle gene, and they produce a GP33 and 34. They are special sigma factor for the lead genes, all right, and to produce lead protein. So this is alternative sigma factor when the bacteriophage infect the bacteria, so that they can use the bacteria, the host, the coenzyme, and then to produce the viral gene and viral protein, okay? That's one strategy. The other one is RNA polymerase switching. So this happened, for example, the phage T7, okay? Phage T7, okay? They have 3 phases of transcription. So they have the class 1 for early gene, and then class 2 and 3 will be late phase, all right? Instead of making the new sigma subunit, the class 1 gene, okay, they form the new RNA polymerase. So that means complete sigma factor and coenzyme. Here you will see the new sigma factor, and this is bacteria coenzyme. But in here, both coenzyme and sigma factor are completely new. So that'll be the polymerase switching, okay? And then anti-termination. So anti-termination basically also when the bacteriophage infect the bacteria, so they use these terminator. So for example, origin they have a terminator to control the transcription, all right? The bacteria, they want to defend the viral infection. They will terminate their transcription. However, there's the anti-termination protein, okay? They will come in and then to help the RNA polymerase to continue to process the transcription, okay? So that's anti-termination, so that they can read through this terminator. So this is the example. So the lambda phage, okay, infection. One of the examples here that's during the lytic cycle, all right, all the phage gene can be transcribed and translated. In lysogeny, okay, in lysogeny, basically, they will have the lambda repressor protein expressed, and then they prevent the transcription of all rest of the phage gene. And then when they are going to switch to the lytic cycle, all right, this is the lysogeny cycle. When they want to transform to the lytic cycle, then they have the anti-terminator. They produce the anti-terminator. So at this stage here, right, so they live with the bacteria together. But when they switch to the lytic cycle, then they have the anti-terminator, so that they can transcribe, immediate early, delay early, late gene, so that they can switch to the lytic cycle, okay? So that's the M protein, that's the anti-terminator. And operon, that's another regulatory strategy, okay, regulatory strategy. So operon, basically in the prokaryotic cell, for economy reason, normally one promoter can control several genes, and these several genes, a group of genes, they involve in the same metabolic pathway, and this group of contiguous coordinately controlled gene we call the operon, okay? And the expression of this gene can be triggered by the presence of a subtle substance we call inducer, and this phenomenon we call the induction, okay? And so So because all the genes in this operon are transcribed together to produce one message, we also call this as a polycystronine message. Polycystronine message. Okay, the polycystronine message, and there are several types of regulation. Okay, so under the negative control, So basically, under normal circumstances, right? So the repressor will be produced, and then they bind to the promoter, and then you have repression, right? The co-repressor bind to the inactive repressor and bind to the promoter, and then you have repression. And then the negative control induction. So basically, the repressor will bind, right? If you didn't add anything, they will bind. If you add something that's co-inducer, that will change the conformation of repressor, so it become inactive, then it will be induction. That's co-inducer. All right, for positive control, all right, you have the inactive inducer. If you have a co-inducer, then it become active. If you remove the co-inducer, then it's inactive. All right. And here for repression, the active inducer, if you add a co-repressor, then you will not bind. All right. So it's a repression. So that's the basic regulation. All right. So the detail here, so for example, the negative regulation versus the positive regulation. Okay. So for the regulation, for the positive regulation, if you look at positive regulation, okay, binding of regulator to the DNA. If there's nothing bind, the expression is off. If you bind, then it's on, all right? So there's positive regulation. If there's no regulator, no transcription. If you have regulator, you have transcription. So this is positive regulation. Negative regulation, okay, if there's no binding of a regulator, then it's a transcription. If you have binding of a regulator, it's off, that's negative, all right? So here is a negative, and this is a positive. Okay. So for this example, the leg operon, negative control of the leg operon, okay? So when there's no lactose present. So the leg gene, all right, they produce this repressor, and then they bind to the operator, which should be overlapped with the promoter. Therefore, the polymerase will not be able to bind to the promoter, so no transcription. So in the absence of lactose, right? In the absence of lactose, then you add the lactose. Okay, you add the lactose, you do repression, because lactose will be the inducer. Lactose will be the inducer. They will bind to the repressor. So the repressor will no longer able to bind to the operator. So the polymerase comes, so you have transcription. Okay. And then another one will be the positive control by the CAP. Okay. CAP basically, the both glucose and lactose are present. The cell does not make the lac protein. Okay. The repression of the synthesis of the lac protein by glucose is called a catabolite repression. That involves the two regions of the promoter. One is the binding site for the RNA polymerase, which is a promoter, and the other one will be binding site for the another regulatory protein, we call the CAP. Is a catabolite activated protein. Catabolite activated protein. So when the glucose is not present, the CAP form a complex with this. All right. When there's no glucose, the CAP bind the cyclic AMP. So you have a cap cyclic AMP complex. No glucose, then this form complex, all right, then they can bind to the promoter. Once they bind the promoter, and also in the presence of lactose, they can help the RNA polymerase to stabilize the promoter so that they can have the transcription. However, in the presence of glucose, all right, in the presence of glucose, this level, cyclin AMP level is very low. Therefore, they don't have enough complex to bind to the promoter, and therefore they cannot stabilize the polymerase, okay? And so they will have a very little transcription. Okay, so that's the CAP side. That's an example of the enhancer too, yeah. And then the transcription attenuation. So basically attenuation, so like the trip-operon. The trip-operon is the operon to produce the tryptophan, okay, the amino acid tryptophan. The trip-operon, basically they respond to a repressor protein that bind to a molecule of a tryptophan, okay. When the tryptophan is plentiful, so the repressor tryptophan complex bind to the trip operator, and then no transcription. So when the tryptophan level are reduced, then the repressor will not bind the operator. So the operon is transcribed, okay? So this is a repressible under negative regulation. So this is a standard negative regulation. And also, apart from this regulation, the trip-operon operon can also be regulated by the transcription attenuation. So basically, the mechanism operates by causing the premature termination of transcription of the operon when the tryptophan is abundant. All right. Basically, they just have they have two loci. We have trip leader, right? The trip leader, all right, and the trip attenuator. Okay, these are in the between the operator and also the gene trip E, right? So basically, if the tryptophan is plentiful, then they will form the secondary structure. They will stop the polymerase, right, RNA polymerase to go through the attenuator, right, to enter the trip E. So that lead to the early termination. That's transcription attenuation. Okay. And ribose switch. So basically, just about the 5' untranslated regional messenger RNA. Okay. They can function to regulate the transcription. Okay, so that's basically transcription system in the prokaryotic cell. For the eukaryotic cell, okay, as I mentioned earlier, there are 3 different types of RNA polymerase. We have 1, 2, 3 different polymerase. Okay, polymerase 1, they synthesize large ribosomal RNA, including 28S, 18S, and 5.8S ribosomal RNA. And polymerase 2 synthesizes messenger RNA and also these heterogeneous nuclear RNA, all right, and small nuclear RNA. Okay, and then polymerase 3 make the precursor to the tRNA, 5S ribosomal RNA, other small RNA. Okay. And then again for the polymerase, okay. So for the polymerase, remember for the polymerase prokaryotic cell, we know alpha, beta, beta prime. So for the eukaryotic cell, they have this RPB123, which is similar to the beta prime, beta, and alpha subunit, okay. And all 3 eukaryotic RNA polymerase, they are large complex. They have 10 or more subunits. But these are 3 fundamental subunits they have, which is similar to these beta prime, beta, and alpha. Okay. And so promoter region. So different type of polymerase need a different type of promoter structure. Okay. So if class 2, like we mentioned, class 2 is similar to the regular RNA polymerase in prokaryotic cell. So of course they need a core promoter. All right, the core promoter. Core promoter is like this with Tarabox -25 and initiator, all right, downstream element and upstream element. So these are just a co-promoter, all right? And proximal promoter, basic proximal promoter is upstream of the class 2 promoter. And then the proximal promoter, so basically this co-promoter is bound by general transcription factor. And then the proximal promoter, basically that would be bound by the specific gene specific transcription factor that's transcription activated depending on the gene sequence, okay? And then class 1 promoter. So a class 1 promoter is now well reserved in sequence from one species to another. They all have similar general architecture of the promoter because they have a core element surrounding the transcription start site, and also an upstream promoter element. Upstream promoter element right here. Upstream promoter element, which is upstream control element, right? It's 100 base pair for the upstream. And then we have core elements surrounding the transcription start site. Okay, these are two elements that are typical in the RNA polymerase 1 promoter. Okay, and then class 3, there's 3 different types of class 3 promoter. Okay, so the class 3, so the type 3 here is similar to class 2 promoter. And this class, the type 1 and 2, basically they have the downstream sequence that can be recognized. Okay, they have this downstream internal promoter. See the transcription start site, then the box A, box C, right? This internal downstream internal promoter. That's for the class 3 promoter for the RNA polymerase 3. Okay, and then for the order of event during the transcription initiation. Again, of course, the transcription, you have initiation, elongation, and termination. For the initiation, it's very special for the eukaryotic cell because you need a general transcription factor. All right, sometimes you need a gene-specific transcription factor, which is activated to help, right? And then for the transcription factor, general transcription factor, they basically combine with the RNA polymerase to form this pre-initiation complex, right? So for the class 2 pre-initiation complex, we have RNA polymerase 2 and then 6 general transcription factors that involve in the initiation, A, B, D, E, F, H, all right? And here will be the process, okay? The process of the transcription initiation. So first of all, we have talabox, right? Talabox, then the TF2D, the transcription factor 2D, right? Recognize the talabox. So they will bind to the talabox with the help, right? TF2D bind to the talabox, all right? With the help of the TF2A, all right? And so they form this DA complex. And then B then would join. So they form this DAB complex. The TF2B is critical for the assembly of the initial complex, all right? And also for the location of the correct transcription start site as TF2B, all right? And then after that, after they form the DAB complex, then F will bind to the F bind to the RNA polymerase 2 and then bind to the promoter, okay? So basically F would bring the pol2 to this DAB complex, okay? The F would bring the pol2 to this DAB complex. They form this complex, all right? And then EH finally would join, all right? Join, and then they form this pre-initiation complex. Okay, and then to start the transcription, and then the RNA polymerase 2 will be phosphorylated, all right? Will be phosphorylated, and then they can start the initiation. Okay. So that's the order of the event. So the D, A, B, F, and PO2, and then EH, right? And TF2D, basically, they have the Tara box binding protein, so-called TBP, and also TF, Tara binding protein associated factor. They help the TF2D to bind to the promoter, okay? And then the transcription activator, basically the gene-specific transcription factor depend on the different type of gene that you want to transcribe. You need different type of activator. And then activator, normally they have two different domains. One is called activation domain. Activation domain basically help to stabilize the RNA polymerase. And the other part of the transcription activator is DNA binding domain, because the DNA binding domain will come to recognize the proximal promoter and help to bind to the promoter. Okay, so for the structure of the activator, basically the DNA binding domain, they need a binding motif. Okay, motif. We have the zinc containing module, which is a zinc finger. The zinc finger, which is most common seen domain for the transcription activator. Okay, it can help to bind to the DNA. Also, we have a homeo domain, right? The zinc finger, homeo domain, and then the BZIP and BHLH motif in the transcription activator, so they can help the polymerase and help the activator to bind, and then help the polymerase to stabilize the promoter. Then from the prokaryote, right, the gene regulation, we learn those several different strategies in the prokaryote cell. For the eukaryote cell, okay, there are two major regulation, right? It's epigenetic regulation, because that involve the nucleosome, right? Involve the nucleosome. Remember that we learned that the DNA wrap around the nucleosome, right, in the genome. So basically, the DNA basically is buried into the nucleosome structure, and if they are buried inside the nucleosome structure, the RNA polymerase transcription activator will be impossible to access to those DNA binding site. All right, and if they are not able to access the binding site, they cannot initiate the transcription. In order to make the DNA available to the transcription activator and the general transcription factor, we have to remove the nucleosome so that the DNA will be available. All right, and there are two mechanisms that we can perform this job. One is the chromatin remodeling complex to remodel the chromatin, and the other one is histone modification. Okay, histone modification. This is a picture of the nucleosome, and inside here is the cohistone, and this is DNA, and this is histone tail. All right, so for the first one, chromatin remodeling complex, basically they will be able to remove the nucleosome from the promoter temporarily, okay, either by the octamer transfer temporarily or octamer sliding, so that they can make the DNA available for the transcription factor to access. And the other mechanism, epigenetic mechanism, that would be the histone modification. As I mentioned earlier, that the histone tail, positive charge, DNA is negative charge, so they interact with each other, right? And in order to make the DNA, okay, flexible, you have to neutralize the histone tail. Neutralize histone tail, I mean those lysine arginine residue should become neutral, not positive, right? Once they become neutral, they will no longer able to bind to the DNA. And that process is called histone acetylation, right? So histone acetylation basically will be able to let the DNA available for the transcription factor. And from all this research, the histone modification and remodeling complex, then we have these terms called histone code. Histone code just to give the signal for the transcription factor, whether they can be transcribed easily or not. That's called histone code, because they don't change the DNA sequence, they don't change anything. It's just that the nature of the histone has been changed. So, for example, the histone been transferred, been remodeled, and the histone tail been neutralized, and for low signal that can allow the transcription factor to access, and then you'll be able to transcribe that particular gene. That's called histone code. Okay. So we will watch this briefly.
Epigenetic regulation encompasses a number of different modifications to chromatin. These include methylation of the DNA on cytosine bases, a modification that can further be oxidized, as well as modification of the histone tails that emanate from the core of the nucleosome. The tails of core histones labeled here can be altered with distinct chemical modifications, including methylation of histone H3, acetylation of histone H4, and phosphorylation of histone H2B. Euchromatin is often characterized by a more open and accessible state of the DNA, one in which transcription factors have access to their cognate binding sites and can therefore recruit enzymes like histone acetyltransferases that acetylate histone tails and activate genes by recruiting components of the basal transcriptional machinery, including RNA polymerase. Heterochromatin, in contrast, is thought to be characterized by a more repressive, tight bundling of nucleosomes, which impedes transcription factors from gaining access to regulatory sites on the DNA. Methylation of cytosine bases in regions called CPG islands is a hallmark of transcriptionally repressed heterochromatin. These methylated cytosines in turn recruit proteins like MECP2, methyl CPG binding protein 2, and HP1, heterochromatin protein 1. These proteins are thought to maintain a repressive state of chromatin by inducing histone deacetylation by HDACs, as well as histone tail methylation by histone methyltransferase enzymes.
Okay, so this is the typical epigenetic control, the regulation of transcription. Okay, and so as we learned that in the eukaryotic cell, all right, so the after the transcription, what you get is called pre-messenger RNA, and then the pre-messenger RNA, okay, need to be processed, right? Okay, you need to have the post-transcriptional modification, and so that the pre-RNA will become the mature messenger RNA, all right? So in here, as you can see, the gene, eukaryotic cell, we have the intron, exon, intron, then you transcribe, all right? So after the transcription, you have this pre-messenger RNA. The pre-messenger RNA, you have this 1' untranslated region, 3' untranslated region. Then you have to go through the processing. So that become the mature messenger RNA. And then this mature messenger RNA will be transported to the cytoplasm to the site of the ribosome for the translation. Okay. So three important process is capping, polyaddition, and the splicing. All right, the capping, that's a capping, poly tail, and splicing. All right, remove the intron, combine via exon. Okay. So the 5' capping, first of all, 5' capping occurs soon after the growing RNA emerges from RNA polymerase II, right? So basically accomplishing three steps. First, RNA 5' triphosphatase, right? They catalyze the removal of one phosphate from the triphosphate at the 5' end of messenger RNA. And then the guanine transferase, then attach the molecule of guanosine monophosphate, GMP, all right? So first step, you remove 5' phosphate, and then second step, add GMP. And third step, you methylate the guanine 7, right? So that's a capping, all right? The capping basically can protect the RNA and also guide the RNA to the ribosome, okay? The methylated guanine here, okay? That's a 5' cap. And then the second step will be the poly A tail, all right? Poly A tail, basically the poly A adenation of the 3' of the eukaryotic messenger RNA begin with the initial cleavage of the messenger RNA. So the cleavage, basically, you have to cleave the RNA, right? So occur after the CA nucleotide pair. The CA nucleotide pair, where is this? The CA nucleotide pair basically lie between a conserved AAUAAA, all right? AAUAAA hexamer, and also a UGU rich region. In between this region, there is a dinucleotide CA. They find this dinucleotide CA, then they will cleave this RNA. All right, so they remove this part, and then you have the polyadenation. Okay, by the poly A polymerase. So you add poly A. So the poly A is about, normally it's like a 20, approximately 20 adenosine are added. Okay, that's a poly A tail. And then the third step would be splicing. Okay, so the splicing, because the mature transcript for many genes is encoded in the discontinuous manner, we have to remove the intron, connect those exons together, right? Connect the exon together. And so basically, the splicing, okay, the splicing is carried out by the complex we call spliceosome, okay, spliceosome. Spliceosome basically is composed of several individual small nuclear ribonuclear protein, and also many other protein, okay? But mainly those are small nuclear riboprotein, okay? That form this spliceosome. And the spliceosome, basically, they have this 5 small nuclear RNA, all right? So that's U1, U2, U4, U5, U6, okay? These are small nuclear RNA. They are post-transcriptional modified protein. And this snRNA, they can recognize the 3 sites on the pre-messenger RNA. So we have 5', so these are the spliceosome, they form the spliceosome. And then they will find the 3 sites in pre-messenger RNA. So we have 5' splice site, okay? That's where that use the 5' splice site, which is here, right? 5' splice site, and 3' splice site, which is here, and also the branch site. So those spliceosomes, this subunit, they bind this area, okay? You see the U1, right? That's U1 snRNA, and then we have this, the U2 AF, right? U2AF, which is a U2 accessory factor, okay? And then we have 5' splice site, 3' splice site, and then with branch site as U2, all right? U2 bind to the branch site, okay? Remember, U1 bind to the 5' splice site, U2 accessory factor bind to the 3' splice site, U2 bind to the branch site. And then U4, 6, U5 come in, all right? They bring this together, all right? Bring these together, so this intron, all right? They form a loop, all right? Form a loop, and then they cleave the 5' splice side, all right? Then this point, then they will form a loop and connect to the branch side, all right? Branch side, and then the 3' splice side will be cleaved, all right? Will be cleaved, so that you remove this part. you remove this part, and then you connect from here to here. So that's what you see. That's called splicing. So this is splicing process, okay? So basically the intron and exon are cut apart at a 5' splice side, all right? You cut it here, so you free this end. And this end, then they will join to this the branch side, okay, to form the loop. And then we'll cut a 3' splice side. So then you free this 3', so you can remove this part. And then they rejoin this part to this part here, all right? That's the splicing, okay? And the majority of the messenger RNA use this method to splice the messenger RNA. In the organelle, like the mitochondria, chloroplast, or lower eukaryotic cell, they use a self-splicing mechanism, which is different from this, but that's only a very little use that mechanism. But the majority, they use this spliceosome to perform the splicing, all right? So that's how they become the mature messenger RNA, okay? And then alternate splicing that can create this protein diversity. As you can see, they can connect different part of exon and then give you different protein, okay? Now let's watch the video for the messenger RNA processing.
Before mRNA can be used by ribosomes as a template for building proteins, it must first be processed. Key steps are the addition of a methylated cap and a polyadenylated tail. Involved in processing are RNA polymerase, cleavage factors, and poly-A polymerase. Processing of mRNA begins with transcription. Soon after RNA polymerase begins transcription, a methylated cap is added to the 5' end.
Remember, that's a guanine,
Transcription
okay?
then continues to completion.
The 5' cap, that's a guanine, okay?
Following completion, RNA polymerase releases the capped strand of pre-mRNA. Specific nucleotide sequences in the mRNA are bound by cleavage factors.
Remember, that's
The 3' end
AAUAA and then poly G, and then in the middle CA, they will cut here and then add the poly A tail.
of the mRNA is next moved into the correct configuration for cleavage. Stabilizing factors are then added to the complex. Poly-A polymerase now binds to the mRNA and cleaves the 3' end. The complex begins to dissociate and the cleaved 3' end quickly degrades. Poly-A polymerase now synthesizes the polyadenylated tail by adding adenine residues to the cleavage site. Additional proteins then bind to the tail, increasing the rate at which it grows. When the tail reaches its full length, the poly-A polymerase signals to stop adding residues, and the polyadenylation process is completed. The processed mRNA is now ready to undergo splicing in preparation for translation.
Okay, so that's the RNA processing. And now the non-coding RNA, right? So 98% of transcriptional output of our genome is comprised of non-coding RNA. It's called ncRNA, okay? And the length of the ncRNA can vary from 21 to several thousands nucleotides. So we have a long non-coding RNA, so it's LNCRNA, or small ncRNA, right? Small non-coding RNA, right? So we have microRNA, is miRNA, okay? MicroRNA is endogenous to the cell and produced by the transcription of the cell's gene. It's about 22 nucleotide long. It's cut from a larger RNA, contain a hairpin loop by the enzyme dicer. And also we have small interfering RNA, and the small interfering RNA normally that refer to the exogenous source, okay, like from the viral infection or the synthetic double-stranded RNA, they can be cut by the dicer to between 21 and 25 nucleotide. That's siRNA. And small nuclear RNA, small RNA, small nuclear RNA, snRNA. Okay, so these are small ncRNA. Okay, and then for the RNA interference, so like the internal, the miRNA and the external siRNA, it can be cut by the dicer. All right, the dicer is one of the RNA's 3 endonuclease. Okay, you would take the larger double-strand RNA and cut them to a small size. Okay, and once the RNA is cleaved, then they pass a protein complex. It's called RNA-induced silencing complex, called RISC, RISC. Okay, this RISC protein, okay, will bind to the RNA. All right. So basically, RISC, then they will unwind. They will unwind these double-stranded RNA, and then they will select an antisense strand. Okay, they will select the antisense strand. Okay, then this nucleus protein, the RISC complex, we call it AGO, AGO subunit. They will guide the complex to the target messenger RNA. They will guide this target because this antisense, right? Antisense, they will target to the sense RNA, which is messenger RNA, all right? And then once they bind to their target, okay, then they will degrade, they will cleave it and degrade it. So that's called RNA silencing, okay? Interfering. Okay, so so basically those the miRNA, siRNA, they can replace the messenger RNA. Okay, they can also trigger the they can also repress the transcription of a specific gene, and also the large region of the genome through the associated with different complex we call RITS, RITS. RITS here, RITS complex, it's called RNA-induced initiation of transcription silencing complex. Okay, so basically the antisense RNA strand within the RITS target the RITS complex to specific gene promoter or large region of chromatin, and then the RITS will recruit the chromatin remodeling enzyme to the promoter, and this enzyme methylate the histone and DNA and result in the heterochromatin formation, all right, and then induce the transcriptional silencing, okay, if you're RITS. Okay, let's watch this video. RNA interference.
Scientists have been making rapid progress in understanding RNA interference, or RNAi. Many organisms use RNAi to control genes, and it can also be used as a tool in the laboratory, and in the future, perhaps as a therapy. This animation will introduce you to the principles of RNAi involving two important types of RNA molecule, small interfering RNAs and microRNAs. Eukaryotic cells have many sophisticated ways of controlling gene expression. In the complex environment of a cell, these mechanisms need to be precisely targeted. There's a group of mechanisms that use small RNA molecules to direct gene silencing. This is called RNAi. Inside the nucleus, most genes that encode proteins are transcribed by RNA polymerase II. The primary RNA transcript is processed by splicing and forms a mature messenger RNA, sometimes called mRNA. The messenger RNA is then exported from the nucleus into the cytoplasm. Here, ribosomes catalyze translation of the messenger RNA to form polypeptide chains that fold into proteins. But this is also where some small RNA molecules can have their silencing effects. There are several types of regulatory small RNA. Small interfering RNAs, known as siRNAs, are derived from longer double-stranded RNAs that are either produced in the cell itself or are delivered into cells experimentally. The introduction of siRNAs, or double-stranded RNA, is widely used to manipulate gene expression. MicroRNAs are another type of small RNA. Most microRNAs come from RNAs that are transcribed in the nucleus, which then fold and are processed before being exported into the cytoplasm as double-stranded precursor microRNAs. The double-stranded precursors of microRNAs and siRNAs bind to dicer, which is an endonuclease protein that cuts the RNA into short segments. Most siRNAs and microRNAs are approximately 21 nucleotides long. The short double-stranded RNA then binds an Argonaute protein.
That's AGO protein.
One strand of the RNA is selected and remains bound to Argonaute. This is called the guide strand.
That's antisense RNA.
The combination of the RNA and Argonaute, along with other proteins, is called the RNA-induced silencing complex, or RISC. siRNAs direct RISC to bind to specific messenger RNAs. The targeting is precise because it's determined by base pairing between the siRNA and the target messenger RNA. siRNAs often have perfect complementarity to their target sites. Once bound, Argonaute catalyzes cleavage of the messenger RNA, which will then be degraded. MicroRNAs also guide RISC to messenger RNAs. Usually only part of a microRNA, known as the seed, pairs with a target messenger RNA. This imprecise matching allows microRNAs to target hundreds of endogenous messenger RNAs. Targeting by a microRNA can lead to messenger RNAs being degraded or translation being inhibited. Argonautes and their small regulatory RNA cofactors are found in plants, animals, fungi, and some bacteria, and their importance in a multitude of biological processes and as tools continues to be revealed.
So that's the silencing, RNA silencing by the microRNA and the RNA interference. Okay, so let's say for chapter 3, the transcription. Okay.
Two families of small regulatory.
Oh. Yep. So that's it for today's lecture in English.